Hello, everyone, and a very warm welcome to our webinar today, Empowering Humans Integrating AI and Robotics for Efficient Drug Design and Discovery. I am Payesh Bahuguna, Marketing and Communications Manager at Iktos, and today's moderator. Let's start with a short introduction of Iktos. Founded in 2016, Iktos is a leading artificial intelligence for de novo drug design company, headquartered in France, with offices in the UK, US, and Japan as well. Our technology is built upon the latest developments in deep learning algorithms for de novo design and AI-driven synthesis planning. And we have a portfolio of 60 plus real-world projects, thanks to our growing team of talented medicinal chemists, computational chemists, AI and ML engineers and data scientists, cloud engineers, software developers, and BD professionals. We have developed several technologies such as Machia, Spire API, Doc AI, and Iktos Robotics that we'll be covering in the webinar shortly. We work on diverse targets and therapeutic areas for a range of clients, including large or medium pharma companies, biotech companies, and research institutes globally. Our speaker for today is Dr. Quentin Perron, CSO and co-founder of Iktos. Before we start, I request you all to send any questions you have during this session in the chat box. We'll be answering all the questions together in the end. All right, so without further delay, let's start today's session. Over to you, Quentin.
Hi, everyone. Very happy to be here today and to present you our work in the field of AI, machine learning, robotics, and how we use artificial intelligence coupled with a robot to run and to automate chemistry and the full process of drug discovery. So, as you may already know, at Iktos, we are focusing all our AI in this part of the full process where for discovering new molecules. As you know, it's a long and costly process. And where we focus our attention is really here in medicinal chemistry aspects, how to discover new hits and how to optimize them from hit discovery to lead optimization, to be able to produce high quality molecules ready for a preclinical candidate. We have developed several technologies which can work together to produce nice molecules and to cover the full process of drug design. First, we have developed Doc AI, which is active learning combined with docking to perform a virtual screening in less than a day on AWS. Using a database of billions of compounds provided by several providers like MQL, eMolecule, WuXi, or Enamine, for instance. It's a very powerful technology to virtual screen at low cost billions of compounds in a short period of time. Then we have Machia, which is our generative AI platform, which generates new molecules in any type of context. It's a complementary approach on top of the virtual screening, depending what you want to do, and we will cover that shortly. Then Spire, of course, designing ideal molecules in silico is one thing, but being able to make it's another thing. And that's why rapidly at Iktos, we have spent a lot of effort developing our own retrosynthesis technology, which is a 100% data-driven technology. Right now, you can have access to it to the web. It's freely available at spire.ai. And the last but not the least, our new robotic platform, where actually the concept is to automate chemistry driven by the AI stack we have been able to build along those years. And this is the topic of today. So the goal of the Iktos Robotics platform is not only to automate chemistry, but also to develop an autonomous lab. The idea is really to mimic what chemists are doing. The chemists are not doing only chemistry. chemists design molecule, think about the roots, and then they go to the lab and they make the molecules. And our vision is really trying to automate that process using AI to imaging the molecules, perform the retrosynthesis, and robots doing that molecule. So again, it's not automating only the chemistry, but the full process. It's important to understand the difference between those two aspects, because automatic, you know, this is like what you can find in a car factory. There is no special intelligence. The system is doing the same task every day. And the goal of an automatic system is to increase productivity. Contrary to autonomous, which make decision, which has a different task every day, depending the to-do list, depending the thing he needs to do. And not only it increases the productivity, but also it solves problem. And this is really the difference versus, automatic arm like this one or a rover on Mars. If we want to make this parallel in chemistry, I would say that automatic, it's more to design library, and autonomous, it's more drug design. So really, again, the goal of Iktos Robotics is to be here on this right-hand side of the slide, building an autonomous system. The key technology to build an autonomous system is actually AI. And as you can see here in this slide, we have recently autonomous car, but actually a car is a very old technology, almost 100-year old. Navigation system, old technology, almost 30 years old maybe. And we observe only recently autonomous car, and simply because AI is a recent technology. And you need AI to pilot a car, because you need to take decision, to recognize environments, and to make decision actually. You have several levels of autonomous. You know, if you want to buy a Tesla, for instance, you will see different level and different price, of course, depending on the capacity of the vehicle to provide these autonomous services. Of course, we are very far from the level 5 today, which is 100% autonomous without where there is no intervention. This is a big difference between all the level and the level 5, is that in the level 5, you have no human intervention. We are very far from that in the field of lab automation. And I think it's very important to keep in mind that human is at the heart of that process of drug discovery, drug design, and we want to keep it like that. It's very important to have that in mind. AI, it's a very powerful tool which allow to do amazing things. For instance, here you have now plenty of AI which transform a text into an image. This is very impressive to see that actually. But if we talk about creativity, it's still a human which input the text here. So human is really the spark of creativity, and then the AI is doing the rest. I like to say that AI proposes, human disposes. So it's really human which is at the heart of the process. So this is a DMT cycle we are currently applying at Iktos. So Machia can generate and design new molecules. Of course, this is operated by a human. I will show you. Then you have Spire, which can be totally customizable for the robot, making sure that Spire is proposing routes which are feasible given the constraints of the robotic platform. We have developed also Ilaka, which is a system which transforms all Spire information into a process which can be read by the robot. So now we are in the make. And right now, the test is performed by external partners who are doing purification, analysis, and testing. Once we receive the new results, we start another DMT cycle. Our goal is really to accelerate this DMT cycle with a minimum human intervention in terms of action. Where we want to have human is in terms of brain and ideas. And it's more when you set up the system, you bring your creativity and your knowledge as a computational chemist or a medicinal chemist. So what about the design? So first, Machia. Machia is a technology we are developing since 5-6 years now. It's a very powerful platform which easily design any type of molecules in any type of context, from heat discovery to late stage lead optimization. You can input data to the system. You can build machine learning models in few clicks, just defining your target product profile. Machia will build automatically predictive models. You can also incorporate your in-house model through external API. And then when you generate molecules using different type of generative AI, we have 4 different type of generative AI, depending where you are in your process in discovery or let's say lead optimization. Machia can take advantage of 3D information, either 3D ligand base, you have a reference molecules, you have no information about the protein, only one active compound. Machia will try to generate molecules with similar shape and 3D pharmacophore. And if you have structure-based information, it's even better. You can upload your PDB file, and Machia will directly generate inside the pockets optimal molecules. Of course, we have access to SPIA, so we have synthetic feasibility, which is embedded into Machia. But also, I will show you, we have designed new generative AI, which takes synthetic feasibility by design into the generative process. And this is this type of algorithm, which are recent technology we have developed at Iktos. It's quite unique approach. It's a template-based generative AI. And the idea is to combine piece of molecules of commercially building blocks around a given scaffold. So here, the goal, for instance, is this is a ranolazine molecules. And we can ask ourselves, are we able to retry this ranolazine molecule starting from this only? So what we do inside Machia, we're going to define one exit vector, another one exit vector, and Machia will be able to search the best templates. So now we have 90,000 templates coming from our SPIA work on retrosynthesis, and we have 1 million building block. And given this initial data, trying to optimize Tanimoto similarity toward these targets, starting from here, can we retry the ranolazine? And actually, this is what we observed here. In 8 steps, so 56,000 molecules, we reach a Tanimoto similarity of 1. So we have been able to retrieve the ranolazine. Versus a classic combinatorial approach, which would require 10 to the power of 12 molecules to find this ranolazine. So this approach is very efficient because it converges very rapidly. It allows the chemist really to set up things as he wants to do. So for instance, here I decided to pick 2 exit vectors, but I could have picked only 1 exit vector. So you can really grow into one direction, grow into 2 directions, but you can also do fragment linking. So you keep the left-hand side, the right-hand side, and you're looking for something new in the middle. So by design, this system allow you to generate very nice molecules, very easy to make, taking advantage of the novelty and the diversity which is available in those providers' building blocks. So it's a very efficient and innovative way to generate new molecules. So here, for instance, I have performed that, and I will show you afterwards on a starting point where I ask Machia to generate molecule in 3D with a similar shape and pharmacophore. And as you can see here, we have molecules having a very nice overlap with the reference molecules. We have the retrosynthesis information here because we use as well SPIA to propose routes. We have the shape, the pharmacophore, etc. So basically, the output of Machia, depending what you are doing, you have, again, different fitness function, different generative AI, but at the end, you have a listing of molecules. So now the question is, how can I go from this listing of molecules to powders, which I can test, actually? So now so from this listing, I need to find roots for each of those molecules. So I need a retrosynthesis technology, and this is where Spire enter into the game. Spire is our own platform, which has been developed, inspired by previous work from Marvin Siegler and Mark Waller. So we use a Monte Carlo tree search algorithm. It's a template-based approach, which is 100% data-driven. And we have worked the last 5 years to improve the technology on many aspects. Right now, again, the technology is publicly available on Spire.ai. You just need to create an account and you have free access during 30 days. Spire is trained on Pistachio database. So we have 5 million unique reactions. A plus B gives C. We can, of course, adapt this technology to your own ELN or to other database of reaction. We have done that several times for several companies. Spire is deployed on AWS and its infrastructure as code, so we can really update things very rapidly, very easily. We have, of course, a database of building blocks. Right now, we have 29 providers, several dozens of millions of compounds. And then Spire, again, use reinforcement learning to find the best routes given the known chemistry and the available building block. Maybe which can make Spire a unique technology on the market is that Spire is full of advanced search options. So you can find routes given all your criteria. You can find routes for a given price. You can define the availability of the starting point. You can define the key intermediate. You can define presence, absence of the structures, define the bond you want to break. So as a chemist, you really can play with the system. You do not just put the molecules, you have roots. You can do that, of course, but you can put molecules and give all the constraints of your project, because we know that depending the chemist, depending your project, where you are, you may have some intermediate on the shelf. You may want to avoid a certain type of chemistry. And which make this technology quite powerful and very important in the technology stack of the robotic platform is the possibility to force Spire to pass by a set of name reactions or to avoid some name reaction. So basically, you can customize Spire to be compatible with your robot and the technology you have downstream. So once you put a molecule into the interface, you have plenty of routes. So usually we stop the system where we found 300 routes. It's a form of stopping, a way to stop the system. And for each of those routes which are fine, you can, of course, have the full tree and see how to make the input molecules. You have the building blocks and the steps proposed by Spire. On each of those steps, if you want to understand why Spire had these ideas to cut the molecules like this, you can click on reference and you can have access to the associated literatures. And Spire tells you, okay, I break the molecules here because in a similar context, in this patent, they have done similar chemistry. So Spire provides the name reaction and also the literature's information. So once you have this information, you cannot send a plain text information to a robot. Okay, the robot needs to have some structured data. So from this text, we have worked very hard to be able to extract this information and to be able to structure it and to recognize the reactant, to recognize the solvent, the atmosphere, the reagent, the catalyst, and of course, in some reaction, you may have several steps and several sequences. So our system works well, but we discover that it's not enough. Because even if you have a very nice system which goes to plain text to structured data, at the end you may ask yourself, okay, do I trust the protocol described in these patents? Did my AI extract this properly? Maybe I have some mistakes somewhere, so maybe I cannot really trust 100% the AI. And I think the most important thing is this protocol described in this patent compatible with my robot? And this is a very important question because, again, robots have some requirements which need to be accepted and actually to be fixed, to make sure that you are doing good chemistry and things gonna work properly. So we have decided at Iktos to develop our own chemistry compatible with the robot we have in hand. So first, we need to avoid very low temperatures. You cannot run minus 75 degrees inside the robot. We need to avoid very reactive starting material, pyrophoric starting material, reactant. We want to avoid also reaction which generates radioisomers, like, I don't know, Friedel-Crafts reaction, for instance. You need to avoid hygroscopic starting material, like lithium chloride or this type of things, which gonna be a nightmare to scale and to add. So we spend time to first select the name reaction we want to run, the most important one of the literatures, and then we carefully study the literatures to find the protocol which are at the same time versatile, which are going to work in a majority of the case, and also which are compatible with the technology. One of the solutions also is supported chemistry, where you can have catalyst and reagents on a supported phase. It's very convenient because at the end, you just need to filter things and usually you have a clean reactions. So we have developed our own chemistry, Suzuki coupling, Mitsunobu coupling, etc., where the only information we need for the chem speed and the robot we have bought is the temperatures, which type of catalyst, solvent, where are they placed on the robots, the sequence, reagent 1, then reagent 2, the solvent, and the duration. And we did that for all important name reaction. Okay, so now we have designed molecules, we have found the roots, and from those roots, we have the name reaction. But for each of those molecules, I have plenty of roots. So which one to select? And this is actually the role of ILACA. For all the molecules I'm planning to be made on the robot, let's say I have 100 molecules to be made. For each of those molecules, I have 300 roots. And you rapidly understand that it becomes quite a complex process to find the roots I want to select. Because as you can see here, from those roots extracted from SPIA, I have the name reaction. From the name reaction, I can match with the template, reaction template I've been able to create. So for each of those templates, I know the duration, I know the temperatures, I know the sequence of name reaction. And from this information, what I want to do is to be able to batch roots together. For instance, these roots, these steps, this one and this one, are run at the same temperatures and the same duration. So I can put them together on my robot and I can run them the same day. I don't really care about the associated chemistry. Here I have a Mitsunobu, here I have an amide coupling. But that's fine. I don't really care about the name reaction. What I care here is that it's the same temperatures and the same duration, so I can run them together. It's a very important concept because we don't want to run classic library. We want to run on the same platform, the same day, different type of chemistry to be able to batch as much as possible chemistry on the platform. And then I can batch the second step of this root X and the second step of this molecule 3 at a different time and sequence and days. But as you can see, we have plenty of scenarios. I have these first scenarios, but I can start with another scenario, which is root 3, and this one, etc. So the question is, which scenario should I take when I have 100 roots to make and 100 molecules to make and 300 roots per molecules? So for that, we have developed a planning algorithm, which takes as constraints of the system the temperatures of the reaction, the duration, and the availability of the building block. Thanks to that, the system is able for each molecules we want to make to find one route, which is the best one for in terms of batching and in terms of speed. The fitness function of this planning algorithm is to make the robot run at full steam, to be full every day. And then once this algorithm has been able to make that decision, we send this information to the chemist. And again, chemists at the heart of the process will be able to review the molecules we want to make, to review the route which has been selected by ELACA, and the chemist has the possibility to say, okay, yes, I agree with that route, or no, I disagree. Even though this route is good in terms of batching and speed, I think it's not really good given this context. So I prefer to remove that molecules. And these molecules will be batched with a different route in another process, maybe the day after or the following day. Once the chemist has validated that, we can order the starting material. So now we enter into the make, and we have decided to move with ChemSpeed, which is a company based in Switzerland. They are known to develop the high quality robots, very robust, and we have been able to observe that since 9 months. The robot run perfectly well without any issue. So the hardware is very, very robust and works very well. This is sold with software, which is called AutoSuite, which is an expert system where you can really control everything inside the ChemSpeed, every position at millimeters. It's the volume, the scale. It's very, very powerful, but it's very complex, and you have a high barrier to enter here and to use the robots. And for us, it was a problem because we have two main objectives with that robot. At the first time, we need to use the ChemSpeed to develop new reaction. So we use the platform as HTE platform, high throughput experiments, and also we use it at a production as a production system to run chemistry for our project. And if you use AutoSuite and on the Monday you want to develop reaction, you develop your workflow, and on the Tuesday you want to use a robot as to synthesize campaign, it's not possible with AutoSuite. You will spend more time setting up the software than to run the chemistry. So we have decided to adapt this technology to our Iktos workflow. And for that, we have developed a software called ELACA. So ELACA, it's at the same time an algorithm for batching things together and finding the routes. It's also an interface. So ELACA for us is more or less the brain of the lab, taking all the information everywhere from the robots and from the design to propose a strategy. In ELACA, you have two interfaces, two possibilities, either to create a new template of reaction. So you're a chemist and you say, okay, today I will develop the mean soluble reaction. I will mix this and this type of solvent, this type of catalyst. So you set up that into ELACA, and then you can run the chemistry inside the robot. Or the day after, you say, okay, I have plenty of molecules to make. I will use the robot now to launch a campaign, and you can go in the campaign items here and to launch all the molecules you need to make given the chemistry you previously developed. So to communicate to ChemSpeed, we have developed a cloud-based platform, cloud-based database, because you cannot connect the ChemSpeed to internet, otherwise you lose the warranties. So we have set up a cloud-based platform, which is in our proprietary cloud, where actually it's at the heart of the system, where in this database we have put all the information regarding campaign, the batch, to start, to stop, the ChemSpeed, the starting materials, the stock solution, the reagent. This database contains lot of different type of items. And once we create a template or launch a campaign, ELACA send the information to the DB. So you fill the DB with all the required information, and then the ChemSpeed executes This each process you have previously developed on AutoSuite previously, and it takes all the information of the DB to run the system. And of course, the ChemSpeed sends the status to the DB because we need to be update where we are into the process. And those statuses are sent to Ilaka. And then once the reaction is done, the ELN is automatically filled by the system. So this is the layout of our platform. So it follows more or less the process of a chemist would do. First, you receive the starting material. So we order pre-rated starting material to external providers. And this ordering and the quantity are automatically assessed and computed by Ilaka. Then when we receive the starting material, we place the starting material here on the plate. The ChemSpeed will take solvents here. So we have plenty of different type of solvent which can be used. And we'll start to fill all the vials. And we start shaking that. And once it's soluble, all the starting material are added inside the iSins. We have two reactors, so two different type of temperature available. And then this heats and you can also cool the plate and until the reaction is over. Once the reaction is over, we take the mixtures and we go here for SPE. So this is for the workup. You can do basic, acidic. You have also metal scavenger SPE. And then you take the filtrates and you put the solution here on top of the robots. It's MTP vials and where you have barcodes, and this is the crude product. Then the crude product is evaporated into a Genevac. We seal it and we send it to external providers. When we want to launch a campaign, it's very easy. The operator arrives in the lab. Ilaka is run to select the best route, but also to check what we have in the fridge today. Because if we don't have a certain type of starting material, Ilaka will propose a different type of scheduling. It's really live, depending what we have in the fridge. Ilaka will output the list of reactants and reagents for to run the campaign of the day. And then the operator just need to take this, fill the rack, and Ilaka will first job will be to check all the vials one by one, to barcode it, to make sure that we have all what is required, and also to memorize position, because we want to avoid human intervention. So the operator can put randomly the vial, and the ChemSpeed will take it and memorize the position automatically. And then, of course, when it's done, it goes to external providers, and we are now thinking and how to integrate in-house purification, analysis, and early ANME testing. So where we are after 9 months, we have made a lot of progress because we have received the robot in March. And the time, to put the robot into the lab, to set up things, it takes some time, but also to when the robot scale 10 milligrams, to make sure you have 10 milligrams. So you need to test also the robustness of the scale, the robustness of the syringe, the pump, and everything. And it takes some time to make sure that the robot is doing what we want it to do. But now we have a fully functional robot, which is connected to our Ilaka technology. Ilaka interface, the beta version has been deployed. So now the chemist can launch a template of reaction or a campaign through the platform very easily. We have run more than 95 campaigns and more than 600 reactions has been launched on the robots. Again, we are not in production mode right now. Those 600 reactions, it's more to develop our name reaction and to develop new chemistry. So that's why we are not at full speed. Full speed will be 100 reactions a day. This is really our goal. And we are not there yet. We are developing the technology. Right now, we have reached 23 name reactions, so protection, deprotection. I think now we cover all the non-cross-coupling reactions, so it opens a very large possibility in terms of chemistry. And we have also amine bond formation, sulfonamidation. So yeah, plenty of reaction. What needs to be done? It's we need to work on the compound management system. Right now, we under that manually. We need to accelerate on that. As I said, in-house purification and early ADME. This is also something we need to develop to make ELACA interface again more and more efficient and with more and more possibilities and a never-ending job. Again, it's to add more name reaction to the system. So as I say again, the chemists at the heart of the platform in terms of designing, in terms of computational chemistry, how to set up the generative AI, what do I want to explore, in which position, which molecules should I want to make and why. So again, the knowledge will be ever coming from the chemist. But we want to transform a chemist which is doing everything, the design, the synthesis, the purification, the analysis, etc., etc. We want to have more a chemist which can pilot some part of the process using a computer, using robots. Maybe not 100% of the process, maybe for late stage lead optimization with very complex chemistry, it will still go to the bench, but for 80% of the process can be run inside in silico and with robots. I think also it's important to distinguish the myth versus the reality. We believe that a lab without human, we are not there yet. Some action right now, I'm talking about organic chemistry, maybe it's less the case in biology, but organic chemistry, some action are faster with human for sure. People say that you can go from target to a preclinical candidate without human intervention. We believe it's not possible. Again, we need the brain of the chemist, of the computational chemist, of the biologist to work all together to design innovative compounds. Any type of chemistry, any type of design, no. In a lab, you have some limitation. You need to define the chemistry you're looking for, to define the design. And this is actually where the AI is mandatory when you have a robot. It's because you can really explore the chemical space in a very efficient way because the AI knows how to explore the chemical space given the constraints of the chemistry. And so you can explore with very innovative molecules, with simple chemistry, with very complex building blocks that the provider offers. And combining those complex building blocks, efficient chemistry, you can design very innovative and very nice molecules and very fast. So as I say, our platform is currently under development. We hope to reach in Q2 2024 full speed of the platform in order to use it at the same time for our own research, but also to propose this technology for collaboration. Right now, we use the platform, so we develop the name reaction, we run some proof of concept, but also we use this platform for 2 in-house projects, and things are going pretty well. And for one, we are only at the beginning of the process, and for the other one, we are in lead optimization. So a rapid use case I wanted to share with you. It's very fresh, very recent, but at Iktos we are working in also we have developed capabilities in Dell. So all machine learning associated to Dell. So we have developed denoisers technology to remove the noise from the Dell to identify the hit. So I will not enter into that right now, but we have developed this type of capabilities, and inspired by this paper, we say, okay, can we use also the platform not only to design new molecules, but also to perform resynthesis of Dell hits and exemplification. So from this paper, they have identified some hits, and the goal was to use the platform to resynthesize some hits, but also to design new ones. Because when you have some hits from a DEL, maybe you want to explore the chemical space around those DEL and to find potentially new ideas. And this is what we did. And we have been able to generate new molecules around the hits found in this paper, having the same shape and the same pharmacophore. To make those molecules, we need two reaction steps. First, a reductive amination, followed by a Suzuki coupling. And I will show you here through the video. Just let me know if you think properly. How I did that with our technology stack. So I went to Makia. In Makia, you can input reference molecules coming from the literature. So this is these molecules. And Makia will generate plenty of conformers. And you select the best one, the one you like, given either the energy or maybe your understanding of the target. And then here I use the fragment growing and I presented previously. And I obtained plenty of molecules combining building blocks with the chemistry, Suzuki coupling, and reductive amination. So I click on this one, for instance, and I can see in 3D the superposition of my generated compounds with the reference molecules, which is on the right. For this generated compound in the bottom here, I have all the information, synthetic feasibility, the pharmacophore, the shape, etc., and also Makia tells me which building block it has used to generate that molecule. So I have at the same time the chemistry and the building block which has been used to generate that molecules having the shape and the pharmacophore I'm looking for. Then I can click on Spire and ask for to make some routes to that molecules. So Spire is able to propose 300 routes to make that molecule. So you have plenty of chemistry you can use to make that one. But the one which is important for me, it's chemistry I can run in my robot. So for that, I go to Advanced Search. I set up an Iktos Robotics parameters, which 3 reaction step maximum, $1,000 a gram maximum. I don't want to have radio selectivity issue. And I can put here all the name reaction I have developed right now in my lab. And given those constraints, I'm looking for routes to make that molecules. And now we have less results, 27 routes. And I have routes actually which pass by steps which are doable in the robot. So here, a Suzuki coupling and reductive amination. And this information is sent directly to Ilaka to be able to run the robots. So in Ilaka, as I said, you can create new templates of reaction. So for instance, here aldehyde reductive amination. Here, this is where we have put in the past our parameters at Iktos. What is a reductive amination is this, and we just set up things. Then here we have all the to-do list, all the 20 molecules we want to make coming from Makia. We can click on details, see the starting points, and see the procedures, etc., all the information the robot needs to run the system. And then I want to schedule these to-do lists. So I click on schedules, start a new schedule, and I have here all the molecules and all the reaction I need to run for this campaign. And I can select each of those molecules and reaction I want to run today. And I have, of course, again, the numbers, the IDs, the smiles, the substrates, which are associated to that, the porters, etc. I have all the associated barcode and all the information required to put that on the platform required for the operator. So we ran that process. As I said, we picked 20 molecules from the Makia design just as a proof of concept. We made the design the 27th, 30th of October, the selection. Then we order the building block early November. We place the order the 7th of November. We resave in the middle November the starting point, the building block. We run the chemistry the 22nd of November. The first step was, as I said, the reductive amination. We had a 100% success rate, and we have re-engaged directly without purification the Suzuki coupling, where we obtained 80% success rate. In average, after LCMS crude, we obtained 50% average conversion overall, which is quite good and completely and enough for purification and analysis and tests, which are ongoing. As you can see, it's quite a recent proof of concept. It was 20 molecules only. If we had run 100 molecules, it would have been the same job. Here is just to test the technology stack. And in just 1 month, we have been able to go from a design into Makia to the crude powder into Spire. And I think here for purification analysis and tests, maybe 2 weeks, 3 weeks are needed. I think I reached the end of this webinar, so I'm quite happy to take questions.